Hello, this is Eva Amson, welcoming you to this Bite-Size Bio webinar, which today is sponsored by Zeiss. Zeiss is an internationally leading technology enterprise operating in the fields of optics and optoelectronics. Zeiss Research Microscopy Solutions is the world's only one-stop manufacturer of light, electron, X-ray, and ion microscope systems and offers solutions for correlative microscopy. Today's presentation is titled How AI Helps Your Daily Microscopy Routine, and is being presented by Dr. Benjamin Maximilian Schwartz from Carl Zeiss Microscopy. Benjamin is Market Sector Manager for Routine Microscopy Applications at Zeiss Microscopy. He studied biology at the Ludwig Maximilians University in Munich, focusing on molecular biology and imaging techniques with submicron resolution. He completed his doctoral thesis at the University of the Federal Armed Forces in Munich and worked at the Department of Aerospace Engineering, developing detection methods for DNA damage induced by radioactive radiation. After completing his doctorate, he gained several years of professional experience in marketing and business. As always, we will have a question and answer session after the presentation. So please type any questions you have into the questions box which appears on the bottom of your screen, and I'll put them to Benjamin at the end. The recording of the webinar will be available at bitesizebio.com. So now over to you, Benjamin, for the presentation.
Hello, everyone, and welcome to my webinar about how AI, artificial intelligence, helps your daily microscopic routine. So my name is Benjamin Schwartz. I'm the Market Sector Manager for Life Science Academia at Zeiss Microscopy. And today, I hope we can talk a little bit about artificial intelligence. At first, some things I want to mention are that everyone is talking about artificial intelligence right now. We have, for example, tools like DeepL that is helping to translate anything with a deeper meaning. We have in the focus of the media, ChatGPT right now that is able to produce texts with astonishing quality. And also there is Midjourney, where we can just describe an image, something that we have in the front of our eyes, and the algorithm is able to produce something out of it. And also there is NVIDIA that is able to track our eyes, for example, at a live webinar. So this is in the public eye right now, and there are a lot of people that say, oh, okay, AI is a fancy word, but this is nothing for the future. This is just a quick hype, but there's nothing coming out of it. And I just want to emphasize that in the last 10 years, I guess, maybe a little bit more even, we have in the industry a lot of applications, for example, that are really crucial to our progress and helped us a lot to advance in the last year. So if you look at this cable car, for example, where the cables nowadays get inspected via artificial intelligence in the matter of minutes and not in a matter of hours or days. We have supermarkets that sort their produce in a certain way that is calculated by artificial intelligence. We have stock trading. We have algorithms for banks, for big banks that basically recognize different patterns and act on them. And in the production of in the producing industry, also we use artificial intelligence to, for example, detect some mistakes in the weaving patterns. So what I want to say here is that this, or a temporary hype, is not excluding an important development. It's just a change of the focus, and therefore it is really a big thing. We are looking at, if we're looking at technology, we were able to collect data since a long time, but we were not really able to use it. And now we are able to exploit the data down to the last piece of information. So this is a great thing, and AI is already and will be a pillar of our progress in the future. So let's talk about the elephant in the room. What is artificial intelligence? So John McCarthy in 1915 coined the term thinking machine. And he said that every aspect of learning or any other feature of intelligence can in principle be precisely described, that the machine can be made to simulate it. So this is something, and he was mainly focused on programming such kind of robots that help in the industry nowadays. And I think to do this in 1955, this is a great thing, because this is a time where TVs looked like that and our glaciers look like this. So I think there is a lot of water running down the river in this time, and he prognosed in this time that there will be a breakthrough for artificial intelligence in around 500 to 500 years, and therefore he created the computer language Lisp, which is up till now still one of the main programming languages for artificial intelligence. And I think when he did this, he did not have in mind that artificial intelligence nowadays is used for face filters. But also face filters are a part of this artificial intelligence cosmos. It's a field of computer science consisting of a lot of applications, a lot of technologies, and in the center of everything, there is machine learning and deep learning. And machine learning and deep learning enable everything else. Like we have natural language processing, which is basically what you have in DeepL or some kind of chatbot. We have speech recognition. So if we talk into our phones, we can send a message later. It includes expert systems, so it's kind of big data analysis that helps us to decide, or the AI algorithm is doing the decision process by itself. We have computer vision down here, which is also really important, for example, for autonomous driving, where a camera is recording the data and the computer is interpreting the distances, the forms, and is enabling us basically to do autonomous driving. Of course, there's robotics, as McCarthy said, or this is the field basically where McCarthy worked in, and the discipline is to not only let robots do what we maybe can't do with this kind of definition or force, and it's also let robotics or let robots do something, experience something, and act on it. So everything in here is artificial intelligence, but a lot of people miss to understand the difference between the different words. So we have artificial intelligence, machine learning, and deep learning, but these are subgroups from each other. So we have deep learning as a subgroup of machine learning, which is basically something in the field of artificial intelligence. And what is often compared or used as the same is machine learning and deep learning. But there is an important difference between these two. For example, machine learning works with limited data sets, and it's able to do specific tasks with data that it was trained on. So this is amazing. But if, for example, I want to train an algorithm that it is able to do a cell detection, and I give the algorithm here, for example, a cell culture image of eukaryotic cells, then it learns the characteristics of the cells by annotations I am doing for, or I show the algorithm how to recognize them. And the output, therefore, is then that the algorithm is able to successfully detect cells. If these were HeLa cells, for example, the algorithm is perfectly capable of detecting HeLa cells. If we compare this to the usage of deep learning, so if I would train this algorithm with deep learning, then we would have something different, because the deep learning algorithm relies on a huge data set, and it's able to learn the means of his tasks. So the algorithm is teaching itself how to perform better and is adding more levels of abstraction in it. This means if I train them with HeLa cells, he learns to interpret the characteristics of the basic model of cells and is then able to give an output of different cell types, morphologies, and also image qualities. So presume that it's a big thing for life science in the future. Why life science and why not every other industry? Of course, every other industry will have an impact, but for life science, it will be enormous. The reason is we are in the beginning of a fourth industrial revolution. So some people say it's because of the internet. Some people say it's because of artificial intelligence, but it will definitely be because of data, because we are gathering enormous amounts of data. And the industries that are leading in this are the chemical, the pharmaceutical, and the biological industries. Why is this like this? Every step in this sectors need a lot of tools, a lot of instruments, and all of these are connected in the means of Internet of Things. They are gathering a lot of data that waits just to get exploited. So let's have a look on the sector in the life science world, and which of these are the best to basically get improved by this AI revolution. So we have, of course, biofuels, a big topic, and also bioplastics, a big topic. They are produced by using fermenters or a lot of biomass, and we can improve the production with artificial intelligence. We have modified crops, for example. So we want to feed the world because the world is growing, or the world is not growing, but we're getting more and more people, and we need to feed the people. So we can improve with artificial intelligence how modified crops are produced and the amount of them as well. For truck development, for example, it's a big thing because to develop a new truck, you have to have a lot of screenings where artificial intelligence can help to identify the right candidate, and which is then used for the further development of this truck. A thing that I want to emphasize here is also insulin production, for example, because since the 70s, we produce insulin with E. coli, I guess, and this whole process could be expensive, and the prices of insulin are in some countries ridiculously high, and with artificial intelligence, this could be a reason how to bring prices down. There's also a lot of demand for AI tools, little AI helpers in the academic research. Why is it like that? With AI, you are able to get the last drop out of your experiment, last drop of information out of your experiment. So a big demand here. Vaccine production, a big thing since 2020. Suddenly everyone knows about vaccines, and we could, with the help of AI, be better prepared for such events in the future. The next thing is antibody production. I just wanted to show it because of this cute little bunny. This cute little bunny was in former times producing antibodies for us, and now we don't need them anymore because there is a lot of microbiology involved, and also AI is revolutioning this sector. Not to forget medical diagnostics, like blood smears or some parasite detection, but we definitely need to keep in mind, and this is why this is here in brackets, we need to keep in mind there is a lot of regulation going on in the medical field, and therefore this is a sensitive topic. All right, so what do all of these sectors have in common? It's routine lab work that gets everything started. So routine lab work is the basis of all of these sectors, and we should have a little bit more a more specific look at routine lab work, especially in life science. What is it? So who is doing this routine lab work? It's scientists, it's PhD candidates, it's lab technicians, for example, or even postdocs. And what are they doing? So what is in their heads? It's lab equipment maintenance, running PCRs, it's molecular cloning, it's pouring agar plates, it's preparing buffers, for example, it's routine microscopy. So what do you think? What is best suited to be improved with artificial intelligence in the future? I would say it's routine microscopy. And the reason is it's based on data. So what is included in this routine microscopy? We have, for example, 2D cell culture. We have 3D cell culture, working with spheres, working with organoids for drug development, for example. We have tissue culture and 3D cell culture, which is, we have medical imaging and diagnostics, again, in brackets, as I already mentioned to you why, but it would improve everything, every diagnostic step a lot to be able to recognize diseases earlier and with more reliably. We have routine microscopy in histology and tissue sections, where we could be able to identify specific structures in tissue earlier or better. We have molecular cloning, transfection efficiency checks, where we would be able to really detect the minimal possible protein expression. And we will have microscopic manipulation and in vitro fertilization, for example, where maybe we could be guided during the whole process. But of course, this is also something that should stand in brackets because it has something to do with the medical field and there are a lot of regulations about it. All right, so let's take a deep dive and really put yourself in the shoes of a lab technician, a PhD candidate, or a scientist, because this is important for the rest of the presentation. So keep in mind, you are working here in a cell culture lab, for example, and all you want to do is you want to get the work done. The time is tight, time is always tight. The experiments are nested together so that you can even do more work. And basically your brain looks like this lab, how this lab is equipped. And your workload consists out of more than I already told you about: experiment prep, maintenance, cleaning. It's also training and teaching. And this can cost a lot of time and a lot of nerves. And therefore, of course, there is coffee. And it's not just for scientists, it's for everyone. Good. So we are, as I told you, in a cell culture lab. In the cell culture lab, the most important thing you do is cell culture. So what is it? We want to grow and cultivate cells so that we are able to do experiments with them. So we start the cell culture, and there are a lot of points in, or a lot of protocols in cell culture that is basically just work that needs to be done. We check for a health status, we check for the confluency, we check for morphology, we check whether there are contaminations, and then this is all cycling and cycling and cycling every day, every week, and every month. And in this cell culture, there are important steps where microscopy comes really handy. For example, for cell counting, so I know how many cells are in my culture. For this checkpoint for the health, and also for cell seeding. So if I think I have enough cells, I have cells for an experiment, then I take them out of the incubator and check them for the confluency and the cell count to see whether they are ready to be used for my experiment. So what is the cell confluency and cell counting? It's pretty easy. Cell confluency is like some cells don't like to be too close to their neighbors, because if they are, they behave a little bit different, and this is something they really don't like. And some cells, they want to be close to the community. They are a part of the cell community, and they thrive if they are in contact with other cells. So you never know how the cells act if you don't know the cell confluency level. You don't know the density in the culture vessel, basically. So how many percent of the surface of the culture vessel is covered in cells, and how densely are they accumulated? This could be also important because if I want to do an experiment and the cell confluency is not right, they can behave different to their normal behavior, and maybe the results are then not reliable anymore. Another big thing, as I told you, is cell counting. So why do I need to count my cells? There are a lot of cells in the culture vessel, so why do I really need to count them? It's easy because if I want to start an experiment, let's say on a Wednesday, I planned an experiment with 500,000 cells. And on Monday, I want to check whether I have enough cells so that on Wednesday I can start my experiment. And therefore, I need to count them. Or another application, if I have an experimental setup like microfluidics, something like this, and I just want to know how many cells do I see right now, and how many cells do I see after I treat the cells. So this is really critical information, and how it's done up to now is a little bit complicated. So for cell counting, I need to add enzymes like trypsin to my cells. I pipette them up and down so that I really get the last cells here in the culture vessel. And I take the suspension and put the suspension, or at least an aliquot of it, onto my counting chamber and count them with this kind of hand counter. So I sit there and I count every little cell here in this microscopy image, and I need to look at all of the cells that are on the grid, that I don't count them double. So this is really not only time consuming, it's also something that is increasing the risk of a contamination, because every time I add something, every time I handle my cell culture, there is a risk of a contamination getting in. I disturb the continued growth. And this is really important because if I want to grow my cells to the number of 500,000 on Wednesday, and I look on Monday and I see, oh no, they need to get, they need to go back in the incubator again so that I really have this count on Wednesday. This is something I can't just do because I detach the cells. So they need time to reattach. They need time to feel happy again, and they need time to get in this growth phase again. So possibility is I have to postpone my experiment, and maybe there was a deadline. This could be, or could have a huge effect. So another thing why this is critical is every time I'm handling myself and putting chemicals into it, I change or disturb the natural behavior of the cells. So if I want them to act normally, or what I assume is normal, then so that the results are reliable, then I should try to handle or add chemicals the less the better. Also, manual cell counting is not exact and it is not reproducible. So if I ask 5 scientists, or would be better if I ask 5 PhD candidates how many cells are here in this culture vessel, I get 6 or 7 answers. So it's really not reproducible. And the same applies for cell confluency. So cell confluency up to now is measured by a kind of educated guessing. It means I look at this phase contrast image and guess how many percent of this image is covered with cells. And I would say here in the first image, maybe it looks like 90%. On the second image, it looks like something around 50%. And if I face reality, then I see here on the left one, it's current view, it's around 68%. And here on the right, it's 39%. So there is a huge difference between the reality and my educated personal guessing. So cell confluency measurement up to date, not exact, and of course not reproducible, because everyone will have a different result for this. So this is why ZEISS is or was developing this AI cell counting and cell confluency tools, the AI cell confluency and AI cell counting modules basically for the LabScope imaging app. And this is amazing because it's just an upgradable feature for LabScope. LabScope is a free imaging app that you can get cost-free on the ZEISS website, and it is extremely easy to use for imaging, for microscopy. And these AI modules are, thanks to a pre-trained and really big data set, extremely flexible to different cell types, different imaging conditions, and it was trained with deep learning technology, and therefore it is able to abstract and interpret the data that it got. So it is not only a really reliable product, it's also something that is easy to use. The focus was exactly on ease of use, and it's a one-button solution. So you don't have to train your algorithm, you don't have to annotate anything. You just have the image and you click one button and you get the result. That's it. This makes it reliable, reproducible, and of course multi-user friendly, so that everyone in the lab is working with the same standard. Let's jump right in. This is what you see in LabScope, then just a short introduction to it. If I click on this little magnifying glass here on the bottom, I get my result and they will look like this. So if you look here, we see the current view is around 55% cell area, and the average. So of course, for statistically relevant results, I need to do this more times, 3 or 5 times at least, and then I get here the average count as well. The same applies for AI cell counting. If you have your phase contrast image here, you see there are a lot of cells. They are really, the manual counting would be a mess, and therefore we just click on one button and we get the result. Here we see the cells in the image are 2,712, and on average. So of course we need to have a lot of or more measurements, more than one at least, and we get 2,900 cells for the whole vessel, for example, or at least for this the average of the images we took basically. So this is an amazing tool, and I would just recommend to you, go to zeiss.com. I can post the direct link in the comments, and I would recommend to you, download LabScope. It's for free. It's amazing for microscopy, and of course for analysis like this. You can also download the 30-day trial version of the AI modules, and you have 30 days to test whether it is something for you or for your lab. Yeah. And what I also want to recommend to you is that you look into the new ZEISS AxioVert 5 microscope, because the AI modules and this microscope work together perfectly. So the AxioVert 5 digital here is an amazing microscope that is able to help with experiments from easy cell culture, basic or simple cell culture, up to more advanced experiments that maybe need fluorescence imaging, and it is small. It fits in every lab, has a really small footprint. You can place it in the dark room, in the broad daylight, also in the wet lab. Microscope doesn't care. You can just take it out of the box. It's easy to install. You just place it on the table, and in 10 minutes you are ready to go. So, and all of this with exceptional image quality. It's reliable. It's future-proof, because you have a tablet holder here, and you can change the tablet with time. So you're never stuck with old technology. And the best of all, it's a one-click solution. So with the push of that button, basically, you could get all the information you need out of your data. It's really an all-in-one cell imaging system. And welcome back, everyone, to the short live demonstration. We see here the Axelrod 5 digital cell culture or cell imaging all-in-one system. And as I already told you, we have here this tablet holder with, in this case, the Microsoft Surface. It is also possible to work with iPad Pro, and thanks to this holder, we are completely bulletproof for the future. No, better to say future-proof, because we won't be stuck with old technology. What we also have here is a really simple slider for the phase contrast, so we can have bright field and phase contrast without the hassle. And additionally, we have here a six-piece objective turret, so we are really variable with the experiments we want to do. We can have 6 different filter options here. So in combination with our included LED light source, we are able to do really complex experiments with fluorescence microscopy with up to 4 different channels. And then there is only the snap button I already showed you, and that's basically it for this microscope. So it's really easy to use. We can connect the tablet via cables, but it's also possible to connect it easily via Bluetooth, and then we can control the microscope even if we're not in the room, for example. So let's change real quick to the AI modules and lab scope on it. So now I have placed some cells under the microscope. These are some simple eukaryotic cells. And what we are doing now is we look for this little magnifying glass here and click it, and then we can choose between AI cell confluency and AI cell counting. And of course, we can do it both together, but I think for demonstration purposes, better to show one after the other. So choose AI cell counting or cell confluency and click just the snap button here, and the algorithm is calculating the cell confluency. And here on the lower right, we see the cell confluency measurement of this area is 48%. Of course, for statistical reasons, we need another scene. We need a lot of different scenes, or at least 3 to 5, I would say, to have really a result we can rely on. So we just take some other areas here, then I will switch again to another area and click the snap button again. Now we have 3 scenes, and we can either open the image here or we open the results. And what we have here is the scene 1, scene 2, scene 3 results, but also the results on average in these 3 images, and the cell confluency in percent as well, which the more images we have should stand for the confluency of the whole culture vessel. So now we will close this, and I will show you how the cell counting works. So I don't want to save this series, and we choose here cell counting instead of cell confluency, and take just this frame, click the snap button again, and we see here the analysis. Of course, we should go a little bit further, And keep in mind, you can also do this analysis with images you already recorded, so you can have a look whether everything works fine, and go back to data you took a long time ago and just reanalyze the data. So let's take the third scene as well and open the results. And what we see here is not just the results for every scene again and the average of the 3 scenes, but also, and this is really interesting, we have here the extrapolated cell count per square centimeter. So this means if we know the surface of the growth surface, basically, of our culture vessel, we can extrapolate everything to the total amount of cells in my culture. And this will be this here. And this is amazing because it's a working step in the lab that costs us at least 15 minutes or 30 minutes if we are not that experienced, or even longer if we have a lot of master students or PhD candidates. This is really taking a lot of time in the daily workflow. So therefore, we shortened this step to not 1 minute. All right, so I think this is what I wanted to show you about how AI can help you in the routine microscopy workflow, how AI is able to make things easier, and I hope that I was able to give you a little bit or interest you a little bit in the AI modules and LabScope, and maybe you go to the website and download the trial versions, download the free version of LabScope, and just try it for yourself. So again, nothing to say then thank you for listening and have a good day.
That was an excellent presentation. We have a few questions from the audience, but before I ask Benjamin any questions, I also have some questions for the audience. And remember that you can still post all your questions in the questions tab, which is on the side of your screen. So if we can have the first audience poll up on the screen. I'll give you a few seconds to answer this one. This should be very straightforward. This is just asking for your job description. And I think once, that's probably enough time for this poll. We can move on to the next poll, which is another question for the audience. This one is asking who in your institute would be most interested in acquiring a new cell culture microscope. And while you guys are answering this one, I will ask Benjamin a question. So one of the things I was wondering is how big was the data set used to train the algorithm for this?
So the algorithm was trained with 170,000 cells, single cells, and different cell types as well, and therefore it is really flexible in what it is able to detect.
Great. And how can you check whether cells are working properly with the algorithm?
Okay, I think this is a great question because I guess in the questions here in the chat, there were some questions like it. If you don't know whether it is something for you, it's something for your lab, you can easily download the trial version and without a risk, just try it out. You just open some images you acquired before in phase contrast optimally, and then let the algorithm run and figure out whether it is working with your cells or not.
Great, thanks. Yeah, I'm just gonna, to follow up on that, I'll go to, there's these few audience questions that are all kind of related, asking if they can use the AI with anything other than cells. So one person's asking about Cercaria cystosomes, and someone else is asking about pollen. So can they use it with that?
Yeah, I think, let me see, for Cercaria, it is something that you just need to try it out. There's a big chance that it will work, or it works to a certain degree. So I think it was, who was it? It was Caroline. Caroline just download the trial version, download LabScope from the Zeiss homepage or website, and try it out. It's a thing of 20 minutes, and then I guess it will help you a lot in the lab. Also, sorry, the pollen question.
Yeah.
So the algorithm is able to detect even detached cells. So in suspension, it is also able to detect yeasts, and I think therefore pollen could work. I don't know whether it's able to differentiate between different kind of pollen in a suspension, but also this is something that you can just easily try out.
Yeah, thanks for that. Maybe we can have a question for the audience again. We've got another poll coming up. So this question is, how many cell culture level microscopes are available in your lab? And while you're counting microscopes, I've got a question for Benjamin again. So here's one of the questions that's asking, was the training done with nuclear dyes like DAPI?
The training was done with annotations, so not with ground truth, with fluorescent dyes. No.
Okay. And how many cell types can be used for confluency counting experiments?
Since we're using the deep learning algorithm, it's not really limited to a specific number, and as well as the other answers, it's the same. I would just try it out.
We also got a few questions basically asking about different types of images. So can we use this with bright field, fluorescence, histological staining? What are the limitations, if there are any?
I would not say that there are specific limitations for the algorithm, since it is able to do some things that we do not really know yet. Therefore, it's not that easy. But of course, there is the intent to have it adapted for fluorescent images as well, if it's not working already.
Let's go to the last poll. I think we've got one poll for the audience left. This is, again, something on screen to answer about who would decide on how to buy a new microscope in your lab. And while people are looking at that, there's also a question about what about stem cells, and I guess any other type of cells that grow in colonies or aggregates? Is it possible to distinguish cells in that? Yeah.
So it's not relying on the outline of the cells. It's basically, this is what I thought about it. It is more likely to detect some inner structures, and therefore should be no problem with stem cells as well.
Sorry, I'm just scrolling through because I haven't been going in order through the questions. Can we use LabScope with images only acquired through the Zeiss microscope, or can you use it on existing images, I guess?
Now that's also a good question. Of course, it works best with a Zeiss microscope, of course. But if you have images and you want to try it out and they were acquired with another manufacturer's system, then of course it will work as well.
And has this approach been used to quantify cancer cells, and what was the outcome of that? Because I guess these are also cells that can be quite irregularly shaped.
Yeah, if we talk about HeLa cells, this is something that is really not that uniform. This is no problem, I guess. If you have a primary culture of some cancer cells, this should also work, or at least it's worth to try.
Yeah, I think it's always worth to try things if you're not sure. Have a look. We've gone through a lot of these already. There are a lot of very similar questions about cell types and image types. Oh yeah, would you? I mentioned histological staining, but I kind of skipped over the second half of that question. Would you recommend using this for nerve tissues?
It depends. It depends on the staining, but I guess there is the algorithm coming to its to the end of its capabilities. I can't say for sure that there is something coming out of the analysis that makes sense. Would be interesting to test, of course.
Yeah. And I think we kind of touched on this earlier, but is the morphology important for detecting cells?
No, then morphology is not really important, because, as I already said, the outlining is not the main differentiator between cell or not cell, and therefore it works with all the different morphologies. It's really identifying and detecting structures inside the cell.
Great. I think most of the questions have already been answered in some way or another, unless something new comes in right now. So I think I'm going to move on to my last question for you. And that is, can I use the AI modules for data analysis only, so with images that you already have?
As I already said, it's perfectly fine to take images that are in storage for some years, and maybe you have data and you were always a little bit too annoyed to count all the 200,000 cells in them, and therefore maybe just get the old data out of the shelf and try it out. Yeah.
Okay, well, thank you very much. And that brings us to the end of the webinar. And on our landing page, you can find some more information, including also the link to the 30-day trial that Benjamin mentioned. So thank you again, Benjamin, for a very illuminating presentation and a great discussion. And thanks to our sponsor, Zeiss. And of course, finally, thanks to you, the audience, for taking the time to attend and listen in. So until next time, good luck in your research, and goodbye from all of us at Zeiss and Bite Size Bio.